postdocs together at NIH a while ago. I actually don't remember where you were before that. I want to say McGill?
Yes, exactly.
Oh,
Yep.
wow. Okay, the memory cells still fire. I do know since then you've been faculty at both Pittsburgh and NYU, and the announcement is wrong. She's no longer at NYU. She's back at the NIH, and so she's local, which is why I wanted to put, we had two new faculty joining that lab of parasitic disease up at NIH, and so I invited them both. And Elodie gets the call to be the first speaker here.
I'll talk about one of my favorite things, parasitic worms. Although, as Steve mentioned, I was in the parasitology group as a postdoc. I'm back in the exact same department in parasitology. But I have to say that most of what I do, I would say 3/4 of what I do in the lab is actually on viruses. And it's on emergence of new virus variants. So on influenza, mostly respiratory viruses. Now, of course, like everybody else, working on SARS-CoV-2 or COVID-19. And I also look at, in the respiratory tract, at the microbiome. So I'm sort of interested in a lot of things, always from a genomic perspective. And so here, I've done throughout the years, after I did the postdoc at NIH, I ended up in an institute that was a genomic institute that no longer exists. It has a different name now. It was called Tiger. It's now called the J. Craig Venter Institute. And when I was there, I started out in parasitology, so parasite genomics, but ended up in virus genomics. And so I've always kept sort of both things in my research group. And when I moved to Pittsburgh and then to NYU, the parasite component has always been more difficult to fund. It's a neglected tropical disease, this particular worm I work on. So it's always, there's always less funding, while the virus work has always been very well funded. So luckily I can do both, and I've maintained doing both throughout the years. So one aspect I wanted to talk about today was actually this new work that we've done on looking at glycosylation in the worms. So there's a genomic component, but there's also the glycome component. And we like to do in the lab a lot of different things where we try to collect a lot of big data sets and then try to integrate the information to better understand the biology. And so these parasitic worms I mentioned here are called Brugia malayi, and they're nematodes. And nematodes are, fascinating, and they have fascinating biology. And most people know nematodes through C. elegans. So they know the free living C. elegans. It's used a lot in biology. It's a model organism. It's used in genetics. But the parasitic worms are harder to work with. The genetics is more complicated, but they're very interesting. And so nematodes can infect or parasitize a lot of different types of hosts. So you have parasitic worms in invertebrates and plants. We know in cattle, it can be a big economic problem to have parasitic infections in cattle, and of course in humans. And Brugia malayi causes specifically a disease called lymphatic filariasis. It's also called elephantiasis. And here's a small picture. This is the tamest picture I could find of what it can do. And actually what it does is that the adult worms can block the lymphatics and cause this swelling that you see here. And so one aspect that's really interesting about parasitic worms in general, you have here on the top, some hookworms. And I assume you guys can see my mouse. I'm not using another. Steve, can you guys see my mouse?
Yep, it's good.
Okay, perfect. So hookworms are, well known as intestinal parasitic worms, and the worms I'm mentioning here are the filarial nematodes. And so Brugia malayi is here, and a very closely related organism or species is Wuchereria bancrofti, and both cause what's called lymphatic filariasis. Now, other worms, such as Onchocerca volvulus, are also filarial worms, but cause a different disease. And in this case, it's called river blindness. Now, the reason I'm showing you this picture is to show you the lifespan that these worms can live within their infected hosts. And we know that, these worms can live a very long time, cause chronic infections, and they are the cause of major morbidity in the world. And many of these parasitic worms infect what we call the bottom billion, so billion people in the world, in the poorest areas in the world, are actually at risk of infection, and many are infected. Now, one of the issues is that in the case of the filarial nematodes, there are no drugs that can actually target the adult worms. That means what you have here are parasites that can live within their hosts for, up to 15 to 20 years. And, you have to have them die of old age to get rid of them. So there's a big push for drugs that target specifically the adult worms. And part of the work we do in my lab is specifically on this interaction to try to break the interaction between the worm and their host. But another aspect of the filarial worms is that many of them are actually infected themselves with their own parasite. And in this case, it's a bacteria. It's a Wolbachia. And if you work on insects, you know of Wolbachia because they do infect about 70% of invertebrates and have a different type of relationship depending on their hosts. In the case of the filarial worms, it's a real symbiosis. If you kill the bacteria, you actually end up killing the adult worm. So there are studies and some antibiotics that are being used to kill Wolbachia and in the process kill the adult worms, but they're still not used as a mass drug administration approach because antibiotics cannot be given to everybody in the same way. So I will not talk about the Wolbachia-Brugia relationship in this talk, but I wanted to mention Wolbachia because biologically it's super interesting. So an aspect very interesting about these worms too is if we look at the epidemiology of the worms, we actually see that in the countries where you have a lot of parasitic worm infections are also the countries where there's very little of autoimmune disorders. Now, this is not necessarily a direct correlation, but it's an intriguing one. And in fact, it's thought that these worms, because of the things they secrete in their host, they have to maintain this lifespan of, years, a decade within their host. Sometimes the infected individual doesn't even know they're infected. So that what these worms are doing is playing with the immune system of their host. So they're secreting things that are immunomodulating. And so there have been clinical trials using live worm therapy to actually control some autoimmune diseases. And one of the best examples is actually IBD. And it's, inflammatory bowel disease. And they give some eggs of worms that are actually worms of pigs that humans can be infected, but for a short period of time. And they do see lower inflammation in these individuals. But, who wants to take live worms? That's nobody really. And so one aspect of our research is to look at what these worms are secreting to see if we can leverage some of these molecules as immunomodulators. And I'll go into that a little bit. But so what is the immunomodulation? What is it doing? And at the base of this beneficial effect is that these secreted products are in some ways suppressing the immune system. And so here is just a representation, a very simplified schema of the immune system and what, you know, the products or what the worms will be interacting with in the host. And also the effects that you would see of what's secreted, some of the cytokines that are secreted that are either, you know, pro-inflammatory or anti-inflammatory cytokines. And what has been demonstrated is that many of the secreted products from parasitic worms are actually doing this really intricate balance between pro-inflammatory molecules and anti-inflammatory molecules so that they can sort of control the immune environment and make it tolerant for the survival of the worms. And there has been, and actually while I'm talking right now at the LPD where I am at the NIH, this guy is speaking right now and presenting to our department. And Rick Maisels has done most of the studies on secreted parasitic proteins and their immune activities. And he's, you know, demonstrated how anti-inflammatory molecules could be used in autoimmune diseases like type 1 diabetes, multiple sclerosis. I mentioned IBD. But some of these proteins or molecules can be used in a pro-inflammatory way. And we know in vaccination, for example, when you have vaccine adjuvants, you want to push for inflammation so that you activate the immune system. So there is work being done on using some of these parasitic proteins or parasite proteins as adjuvants or vaccine candidates. Now, what I'm focusing on today is actually the glycans, and glycans are sugar molecules that are on some of these proteins. And we know that glycans can actually increase the effect of what a protein does. So they exacerbate the effect. And that's why glycans can be important and are used in adjuvants also. So I mentioned what the worm is secreting, and what we call the fraction of what the worms secrete is the excretory-secretory fraction. So secretory proteins are what is actively being secreted. Excretory can be, you know, coming out from both ends of the worm, basically that are just being shed on a regular basis. And so as we talk about ES protein, so excretory, secretory, there's also the component of the glycan, the sugar moieties on these actual proteins in some cases. Previous studies and some of our studies now are showing that in the ES fraction, so what is being secreted or excreted out by the worm, you have about 850 different types of proteins. And many of these proteins, or I would say 20 to 30% of these proteins are actually hypotheticals, meaning that no function has been determined yet. And they're still unknowns. They're unique to the organism, in this case, Brugia malayi. So there is a lot of discovery that remains looking at ES proteins in these parasitic worms. And that's what we've been trying to do in some ways. Now, the glycans, because they're on the proteins, they can directly interact with immune cells. And here's an example of, you know, different helminth species. So here we have schistosomes, nematodes, Brugia malayi being a nematode, the type of glycan that they actually have, and the human receptors that can recognize these glycans. Schistosomes have a lot of different glycans that are actually unique to those organisms. Brugia malayi does not have unique glycans. They're well-known glycans, but they still play an important role. And so our goal is really to understand what is the glycome from this organism, Brugia malayi, and what it does specifically. And as a first step, this is only a schema to show you the many steps that are required in glycan biosynthesis. And so although you can't identify glycans from the genome of an organism, you can identify enzymes that are important for biosynthesis of these sugars. And so they're often done in a stepwise way to generate these glycans. So here's our two questions in this type of study is, what does protein glycosylation look like in B. malayi? What is the glycome of B. malayi? And does that have potentially implications at the host parasite interface? Now, this study I'll show you with the steps we've gone through is still ongoing. I like to present stuff we haven't published yet. But so I'll take you through our thought process and how we've gone about to identify the glycome in Brugia malayi. And so, no parasite presentation is complete without an actual life cycle, but I've made it alive in this case. So Brugia actually is transmitted through first an insect vector. So it's a mosquito. And in the mosquito, that's where there are 3 different larval stages of this parasite until they reach what's called the infective L3, larval stage 3. And what happens is when the mosquito takes a blood meal from the host, the saliva will have some of the larvae, and the larvae do not necessarily go directly or not injected into the blood. They actually fall onto the skin, go through the wound, and then go into what are called the pre-lymphatic compartment, and eventually make it to the lymphatics. And within the lymphatics, they develop into these adults, and we find them in these nodes, in these little nests that are found in nodes. And depending on where they're located, and it depends on the species of Brugia malayi, you will have either elephantiasis of the lower limbs, which is what happens with Brugia malayi, or the upper limbs, which happens with Wuchereria bancrofti. And then the females will have, shed all these microfilaria that can circulate in the blood and be picked up by the mosquito at the next blood meal. Now, the lifespan of each of these stages is quite different. These microfilaria can survive up to 2 years in circulation within the infected host, while the different stages, the L3, the L4, are pretty short-lived in the host. And the adults can survive, as I said, for up to a decade. Now, one aspect that's interesting is really what happens in the males and females. They have a similar lifespan, but we suspect they play a complete different role in the infected host. And looking at the glycan and the proteins they secrete, we've confirmed some of those aspects, and I'll go into that. Now, all the data I'm showing you have actually been generated by Joe Kusa, who just defended last week or 2 weeks ago in my lab, and he's actually joining my lab at the NIH to continue the work he's doing. And Alex Grote was a PhD in my lab who left a year ago, and I'll show some of the work she's done also on these worms. And Alex is currently a postdoc. Now, the 2 questions I mentioned. Any questions while I'm here right now? I see there may be a question before I move into the details of the experiments. No?
Mark, do you want to go ahead and ask?
Sure. Okay. I was just wondering, are humans the only obligate host for this, or does it go into other mammalian species?
No. So the species malayi goes into humans. There's a Brugia pahengi that can go in different mammals, actually. But the way the life cycle is maintained is actually using gerbils, or these gers. So you can infect different mammals with Brugia malayi. And so it's not maintained as a reservoir out in nature in other mammals. It's mostly in the human reservoir, but you can infect small mammals with it.
So if it could be eradicated in humans, there's a hope that they could actually be eliminated.
Correct. Yeah, if it could be eradicated in the host. So right now, because they're using drugs that will affect have these mass drug administration programs in Africa, where they can kill the microfilarial stage, it prevents transmission. And they've had, low levels of infections because of that. The one problem is that it doesn't kill the adult worms. So they have to maintain for at least 2 decades treatment with these drugs so that you let the adult worms die of old age. And so the idea was that it would be eradicated in the long term using the current drug administration programs, but it hasn't happened yet. It doesn't seem to work.
Thank you.
You're welcome. All right. So here's a little schema of the different steps I want to go through today. And initially, we decided to look at the genome, specifically at the genes that are involved in glycosylation. And for that, we did, some kind of pretty intense annotation of the genes using different tools to identify what would be glycosylation genes or involved in the biosynthesis of glycans. And we identified close to 300 genes in the Brugia genome that could potentially be used for this glycogenome. But then the next step is, what are the glycan epitopes that are presented? So what is the glycome? We have the glycogenome, we have the glycome. And so the other step we want to look at is what is actually secreted by the worm that would carry any of these glycans. And what proteins are actually modified with these glycans within the secretome. And then we did some preliminary analyses on immune cells to determine what are the immune effects both of these glycans and of some of the proteins we've identified. Now that aspect is still ongoing, so I'll show you where we are and what direction we're going specifically on the immuno potential of these proteins we've identified. And an aspect I've mentioned is the male-female worms, and that there could be sex-specific host-parasite interactions that we suspect of these worms. We know they can play different roles. So here you have an example on the left of Brugia malayi. These are the males and these are the females. So the males are much smaller. And actually in these nodules where you have the nests of male and female adult worms, you have the males that are intertwined around the female. And so they're, interacting this way, but they're also interacting with the host. And the worms are actually secreting things that are also restructuring the lymphatics to give them the right environment to thrive in the human host. Onchocerca volvulus that I mentioned was linked to river blindness actually has a different way where the male and female interact. So in this case, females form these nodules, and here you can have nodules under the skin, even here under the scalp of a child. And what the males do is that they travel nodule to nodule to fertilize the females. And so we're trying to identify what are these homing molecules that allow this worm to find its way to the next female. And so what we'd like to identify when we're looking at the males and females in these different environments is whether it would be easier to interfere with one of the two sexes rather than try to eradicate both worms, both males and females at the same time with one drug. And so that's why we're interested in this sexual differences between the sexes in these worms. Looking at the transcriptome, so we have the genome we've identified for the glycogenome. We've looked specifically at the expression of the genes throughout the life cycle, and specifically here at males and females. And on the left side, you have your typical heat map. And we see that out of these 285 genes that are glyco-related, we find that 40% are significantly differentially expressed between the two sexes. And here's just an example of some of the genes that fit within these different categories of glycans that we find are differentially expressed in the males and females. And here's just an example that I want to show you from mucin type O, where we see that the differential expression will look like this. So in females, you would see some genes that are upregulated, and in males, different genes that can be, code for similar enzymes, but encoded by different genes. And here you would have a conversion of your serine threonine with the addition of a GalNAc, a galactose, onto the serine threonine. Now that's where things start diverging, is that in the male, you'll have upregulation of two other genes that will lead to the addition of GlycNAc sugar moiety onto that, that whole mix of molecules. And here you'll have also a different addition of, different GlycNAc to form a different structural glycan. And these will have different functions in their interaction with the host, and different functions overall. So this is the aspects we're looking at, and I'm just giving you this as an example. Now, glycans are important because they have information that can be read by lectins, and lectins are actually produced by the infected host. So your glycan on your Brugia or your worm proteins will then interact with lectins that are produced by the host. And so this is why this interaction is important. Now, identifying what are the lectins or what glycans specifically you have. So if you identify the sugars specifically, you can do this by some type of chromatography to decode the structure of the sugars, but that's super complicated. There are people who specialize in that, and they say it's really not an easy thing to do. Another way to do it is to take a whole bunch of lectins, and knowing what type of sugar bind to these lectins, you can then identify what are the glycans through indirectly binding to the lectins. And so a collaborator, Lara Mahal, who used to be in the chemistry department at NYU and who is now the chair of glycomics in Canada, she moved to the University of Alberta a year ago, and she developed this lectin array that she uses a bit like a microarray. So if you're, my age, you've heard of microarrays before. If you're a few decades younger, you may not. But the idea is that you can put on a glass slide these different lectins, and then you can then go over it, you can pass over it your proteins that have different sugars, and the sugars will bind to your lectins. And then you can scan, if you have a fluorophore, you can scan for fluorescence, and then indirectly determine what are the sugars that you have that are binding to the different glycan, to the different lectins. And so here's another schema of the experiment. In our case, we have our male-female worms that we actually will use the lyses, so the whole proteins that we take from lysed worms, and we'll then add the fluorophore, mix with our control. In this case, it's the male-female lysates together are our control, and then our treatment is just either the male or either the female. And then you get co-binding on the lectin if they're expressed in the same way in the male and females, or if they're not. And so here's what it looks like actually. And so on this side, we have our adult males. It looks more green. On the right side, we have our females. And in this particular strip of dots, you see that the color is slightly different. So of course, we don't do this by eye. We do this by scanning, imaging scanning. And through that, we can actually quantify what we identify. And so in this heat map, you have, this is a subgroup from the overall scan of the overall array. And this is just a subgroup of some of the lectins that are found to be bound to differentially in females. So you have in red is highly abundant, and in males. And here I'll focus on two of these specific lectins that we find are binding to sugars preferentially in the males. And these would be the type of sugars that the males would bind to. And in females, it was this one specifically. And so what we were interested in originally was the localization of these lectins, which would tell us where these sugars are in these worms, and why, because we find them in the secretome also, where are they specifically in the worms. So just to show you specifically what these epitopes are, so the lectin in this case that we find is preferentially bound to in the female worms is the GSI, which binds to the GalNAc residues, while this one binds to fucose. So there's more fucosylation in the males and more galactose in the females specifically. So before, I want to show you some imaging that Joe has done with the help of another student. But first you have to see the anatomy of these worms. Now they're not at scale between each other, meaning that the worms are the females are much bigger than the male worms. But it gives you an idea. So in blue here, you would have the intestine, which you'll see in some of the imaging. And in the females, you have the ovaries that are actually towards the end, towards the tail, although the uteri are more central and towards the front end. You have the sensory organs, and they're what would be their mouths, that are at this end. And in the males, you have what's called the spicules right here in the cloaca at the end. And so in the imaging, what we did is that we added fluorescence to these specific lectins. So we have the glycan epitopes, so the GSI that is colored by rhodamine, and the AL with PHTSI. And then we have the male tails, the female heads, and the female tails and the male heads right here. Now to zoom in, so the student from a different lab at NYU who helped Joe was Peter Whitney, who's a whiz on the confocal microscope. And so here the idea is that we know from our lectin arrays and from the glycomics we did that the females have more GSI type, glycans that bind to GSI, while in males it's the opposite. And so the distribution of these glycan epitopes we found right here in the males. You see in both males and females, the AL have a different distribution. So although one, remind you that the GSI was higher in females, there was higher binding to the GSI in females and higher binding of AL in the males, we do see that there's binding to these glycans. The AL is binding to these glycans on the females and in the male, and the GSI the same way, but a slightly different distribution. And this is I was telling you about the intestines, is that's what you see right here. And this is DAPI that is just labeling the DNA in the different cells. And here are the mouthpiece. So you see these different sensory organs in the males and female heads. And so when we do the overlay, you do see that the same organ appear to secrete or have on the mouthpiece the glycans that will bind to GSI or to AL in both. Okay, so these are just the glycans. We don't know what the proteins are. We just know they have proteins with a lot of these sugars, although they're expressed differentially in the two sexes. Now, a different distribution is actually in the tail. So although the mouthpiece, it's sort of similar, in the tail, it's quite different. And this is the male tail, and this is the spicule. And here you see how there's far more of glycans that bind to the AAL in the spicule. And here's an overlay of, I just find these pictures really pretty. That's why I wanted to show them to you today. And these are the ovaries, and you see there is no representation of glycans that bind to these sugars right here. So we see that in the secretion and excretion of these glycosylated proteins, it happens within the mouthpiece. And we see also in the reproductive organs in the male, specifically that spicule, which you can guess what that does. All right, so what you saw,
Elodie?
Yes, go ahead.
So first, you're getting agreement that the pictures are beautiful. I'm glad you did show them. But a question, are you going to try doing any pull downs with the lectins to figure out what the proteins are?
Great idea. That's the next thing I'm going to show you.
Oh, good.
Yeah, yeah. Because when we saw that, we're like, oh, we have to know what these proteins are, obviously, right? And so, yeah, that's exactly what we did. So we saw there was sex bias in that glycome, in that it's displayed in these sensory organs, the secretory organs, the reproductive organs. And so the idea is that what are the secreted proteins that carry these particular sugars, these particular epitopes? And that's when we did, first I want to show you the type of secreted proteins. And so we had done from the spent media, so here nothing about the glycans, it's just the diversity of proteins that we know these worms are secreting. And so we had done this study where we took different stages, so the microfilaria, the different larval stages, adult males, adult females. You can't really culture them, but you can leave them in culture for a few days and then collect the spent media. And that's what Joe did. He concentrated and did protein precipitation and then mass spec. And what was interesting there is this is a representation where you have the list of all the proteins you can identify. So in females, there were a lot of proteins. And many of these proteins are actually unique to the females. So we actually had 238 of all the proteins that were unique to the females, while in males, there were only 6 of these proteins that were unique to the males. So for a total of, 1,000 proteins we could identify throughout these different stages. So this is, much higher diversity of female-specific proteins versus male proteins. So that was kind of interesting that they're secreting different things into their environment. So then, as Steve mentioned, it'd be great to know what, if you take the glycans and you take the proteins, what's the overlap between the two? What is represented with these different glycans? So we did a lectin affinity pull down and then chromatography, and we took worm lysates to do that. We took male and female protein extracts, put them on these different columns, and then eluted off the captured proteins. So the proteins that had the glycans that would recognize GSI or AAL specifically. And so these are just volcano plots showing you fold changes of, what is highly expressed in this case in males. And that would be captured on an AAL. So that would be fucosylated, so captured by the AAL. And then what would be captured by, from the female worms. And you can see that we know that there's more fucosylation in the males, and we see 32 proteins that we are capturing using the pull down from this lectin. While in females, with the GSI, we're capturing a similar number between the males and females, actually. And so if you do, this Venn diagram to see what's the overlap between the types of proteins that we identified in the adult males and adult females, we see that of the 6 found in the adult females, 3 of them are also found in the adult males. Okay, so that's the only overlap we find in these overall proteins we've identified between the males and females. Otherwise, they're pretty much all male specific or female specific, which is interesting in itself. Now, of these 3 proteins that overlapped, there's, 2 that actually are known immunomodulators. So they've been identified before as immunomodulating. So they do have an effect specifically on the host genes and host response. And so the other thing we did is that because we know there are some immunomodulators, one of the first experiments we did is that we took that fraction that we eluted, and instead of doing the chromatography, we decided to take that fraction and test them on PBMCs, which are they're blood cells. It's a mix of different immune cells, and that are from human blood cells. And then to see what effect, by measuring cytokines, to see what effect the fractions have on these PBMCs. It's a bit of a crude assay, but it's to see if there is differential immunomodulation from these different glycan fractions from the males or the female worms. Now, at the time, we had not done the reverse experiment where, we had seen that GSI was higher, the glycans that bind to GSI were higher in the females and the fucosylated proteins higher in the males. We had only done these fractions, and now we're doing the opposite to see if there is a differential effect. Now, the way the experiment works is that you take these cells, they're just cultured, and then you put on it the fraction, and then you test the cytokines and different types of cytokines that I'll show you in a minute. Or you do another type of experiment where you give it a molecule that will induce an inflammation effect. Here it's LPS. So these are, it's a fraction from bacteria that has saccharides, and that actually stimulates an immune response, and that would be your stimulated. So what you're looking at in this fraction is whether your glycofraction will exacerbate that effect or will or down modulate the effect. And so here, to work through these graphs that can look complicated, is that we have 2 different PBMCs from different human PBMCs. And then we also have from each dot you observe in these graphs the first dot represents a control, the control without LPS, and the control here without LPS with LPS. And the second is when we've added the glycofraction. Okay, so what you're looking at here is whether your glycofraction stimulates an effect or maybe downregulates it, but there's not much happening here anyway. So with the LPS, you look at already stimulation, and then you look if there's an effect down or not with your glycofraction. And so what we see looking at these different types of cytokines, so they are representative of the immune response, we see that in some cases, the glycofraction we've put on the cells will, down regulate it, but in some cases will push it up. So it's very complicated what the types of effects that we see. We did see that TNF alpha had an effect in some cases that was quite strong, that we thought could be something to explore further. Now, another aspect is that some of these proteins that we've identified, now, by chromatography, we decided to express some of these recombinantly. But when you're doing recombinant expression, you lose the glycan depending on how you do it, but we wanted to see if some of these proteins have effects. Now, this is even more complicated, but what it shows you, this is a recombinant protein of this particular protein that is known to have a potential immunostimulatory effect. And in this case, all our PBMCs had been stimulated first with LPS, and then we added different concentrations of the recombinant protein at different, microgram per ml concentration, and then looked at whether we saw any effect, what was going on. And so what we saw in general is that actually this protein did have more of a pro-inflammatory effect when you started looking at what was going on in some of these cases. So it was going up, even though they already had been stimulated with LPS. Now, are there questions? I see that there are a few chat questions. I can answer some now.
The chats are still commenting on how great the microscopy was.
Oh, good, cool.
I was just gonna ask that, like, I'm looking at the interferon gamma on this slide, and it, like, two individuals, there's not much effect. One individual, there's a strong stimulation, and one, there's a strong inhibition. Am I reading that right?
Exactly. So depending on these were 4 individuals, we get different effects on the individuals. Yeah, exactly. That's why it's complicated when you're using these human PBMCs. And these were done with healthy individuals. We do have cells that we want to do that have autoimmune, different types of autoimmune diseases. So we wanted to see an effect there too, but we'll need a far larger number of cells or, individual samples to do it on, because there's diversity in the response from different individuals. What we wanted to do first is really identify a smaller subset of proteins we want to focus on. That answers your question, Steve?
Yeah.
Yeah.
Great.
But yeah, you read that correctly. So, as we look at the secretome, the different proteins we had identified that were sex-specific related in the secretome that we had pulled down also from our lectin pull downs, we looked at some of the immune effects of the glycofractions, and now, we're going into some of the recombinant proteins. And what are the immune effects? It's complicated, as you could see. Now, ideally, what we're doing now is that we'd like to have what would be more of a Brugia-like expression system. So when you identify some of these proteins and you try to express them, you're not putting the right glycans on them. You're not doing the right modifications. And so, what we'd like to have is a system where we can put at least some types of sugars on the surface. The recombinant protein work I showed you was actually done, with E. coli. E. coli has no glycans. There's no endogenous like glycosylation. And so what Joe did is that he actually did this massive amount of work where he ended up generating a whole library of open reading frames of all the the Brugia secreted proteins, not all 850, but about 470, because it depends on the size of these. And he put them in an expression vector so we could express them in yeast. And the yeast we're using is Pichia pastoris, which does not have, it has some glycosylation, but it's actually a pretty good system when you're trying to express some, glycosylated proteins or express, purified recombinant proteins. And so that's what we're going through now, when actually Joe has identified, by looking at the transcriptomics and some of the proteomics and the glycomics, he's trying to narrow down the proteins we actually want to use as recombinant proteins to test on our immune assays, to see if we can identify some of these immunomodulators that could be used as therapeutics or developed as therapeutics. So this is, overall sort of the steps that we've taken specifically on the glycome or the glycogenome, and trying to now focus on the mechanism of action, looking at all this. And what we've seen is that there is sex-specific glycosylation, and we think they may be involved in sex-specific roles in their interaction, the worms, how they interact with their human host. And we know that excreted secretory products have immunomodulatory activities, and these are amplified by the glycans. And one last thing I wanted to go over quickly is that I talked about, the proteins they secrete, the glycans on the proteins they secrete, but there's another thing that these worms secrete and excrete, and that's actually metabolites. And so we also did a metabolomics study of these worms. And what we've seen is that actually the males and females may not be secreting the same metabolites either. And if you've done some work in metabolomics, you know that it's even more of a black box. There are a lot of molecules that have never really been discovered. So it's a whole new world. And chemists come up with invented molecules all the time, but molecules that have never been seen in the biological world. So it's always a challenge when you're trying to map back metabolites to any type of metabolomics database. But we tried, and here's an example of looking. So these are the spectra that you see when you do your chromatography of the metabolite profiles. And we did this at different stages. So life stages of the worms, the microfilaria, that larval stage, adult females, adult males. And we did see, more than 2,000 different metabolites. Only 300 could actually be identified. So a whole new world. And we did see, close to 60 that were significantly different between the microfilarial stage and the adult stage. What was interesting is that you do see in the adult worms, these peaks represent specific metabolites. And we did see some metabolites that were unique to the adults. But I wanted to show you this also. In the L3, remember the L3 and the microfilaria, they're found in blood. So the infected L3 are in blood, microfilaria are released through the blood. Now, one of these molecules that is enriched in these two stages is an anti-inflammatory molecule, and it actually is like an analgesic in various animals. So you can see that these worms are secreting molecules that do have an effect to control the inflammation in their host. So it's kind of fun to see this. But in these adult worms and adult females, we did see two different types of molecules. This is one that was, mildly interesting because it had been identified before, but this one we were fascinated by. You can see these two little peaks. And what we saw is that this was a complete novel marker. So this molecule has, you can find in the databases, but it's one of these invented molecules by chemists where, they're playing in the lab, creating these molecules, had never been discovered biologically. And we find that this molecule, this is a volcano plot showing that it's really, really highly expressed by the males, secreted out by the males. And actually we see 2 peaks. So we decided to try to synthesize that molecule. So we went to see our pals in the chemistry department at NYU, and they first synthesized a version of this molecule that was naked. And then they made 2 branched versions. So that means they're adding different things on the molecule. And then what we did is that we did spike-in experiments where you take the chromatography and then you look. So this is the male ES. It's a targeted chromatography where we specifically look at the 2 peaks we could identify. That's our original peaks. We spiked in that first molecule that is not decorated or not different in any way. And this is where it's spiked. So it's actually not these 2. And then we looked at the 2 branched versions and they overlap specifically on these 2 peaks. So it turns out that the male worms are secreting these metabolites that are modified in very specific ways. And actually one thing I didn't go into is that we even did an experiment where we treated our worms with antibiotics. Remember that the antibiotic kills the Wolbachia? Well, we found out that when you kill the Wolbachia, you get twice, 2x higher peaks in our worms when we kill Wolbachia. No idea what that means, but that's what we observed. But we were curious what these metabolites are. So now that we have them synthesized, we can sort of try to understand what they are, and they look like they're short chain fatty acids, they're acetylcholine-like. And so we started looking in the literature at things that have been found to be, antagonists of acetylcholine receptor. And so we were wondering whether this metabolite could affect movement or locomotion. And because it's only secreted by the male worms, we thought maybe it has an effect on the female worms. So we used a very sophisticated assay called the Worminator. It's a motility assay where you put your worms in these cells, in these wells actually, and then you have a camera that will record the movement of these worms. And you can see that the female worms are moving, and then you capture their movement with and without the drug, now our metabolite. So when we did this and we quantified it, we saw that our 2 molecules, when we added them, we did it individually and we added them together because they're secreted together by the males, that they did have an effect on the treated females. So when this is the control, this is a control female. This is the females when we just give the ES product, so the whole gemisch. And this is when we give only our 2 different, metabolites together, and we do see some significant decrease in movement. It wasn't as significant. It wasn't significant in the males. So we still don't know what it does. So we've been actually playing with these molecules, and we've even talked to neurologists to see if we could test them on neurons. And so Bernardo Rudy, who's at NYU Langone, so the med school in New York, and he's actually shown that he tested only one of the metabolites, but it does antagonize one of the receptors in a concentration dependent manner. So this is actually a really high concentration, but we do see an effect on neurons. And this is where we're at right now with all this. So in summary, I've shown you some of the stuff these worms secrete out, but here are the metabolites. There are many novel metabolites that we'd like to explore further. And in fact, that's what Joe will be doing for his postdoc in the lab. We're intrigued by these male or sex-specific, proteins or metabolites that we identify, and we'd like to understand what effect that has on the host and why there would be the sex differences that we observe. And so, we're pursuing further both the protein work and the metabolite of work. So this is the end. I just want to thank, so Kourosh Salehi Ashtiani is at New York, NYU, Abu Dhabi. And he was the co-mentor for Joe, and actually Kourosh paid for all the work on the cloning of the worm proteins into these expression vectors. Lara Mahal, who used to be at NYU, is now at University of Alberta. We did all the lectin work with her. Christine Vogel is a proteomics professor at NYU, and Drew Jones is a metabolomics expert. We still work with all these people very actively now from, we're working from NIH. And this is my lab, some of the lab members. This was our 1980s day, so we're dressed in our 1980s garb, but this is C. elegans with Brugia malayi sitting on it. And here, I put more NYU affiliation because most of that work was done at NYU. And that's it. Questions?
That was great. I'm gonna start. There's Gina. Why don't you jump in? And I don't know if you want, Elodie, you can turn off the share. And
yep, that's exactly what I wanted to do, so I can see people. Good.
I was interested in some of the differences you were seeing in some of the filarial worms where males and females either basically are taking, kind of in the same stationary in the same place versus it's the females that stay put and the males move around. And I wonder if you saw bigger contrasts in some of these metabolite secretions when you have bigger differences in mobility between the male and female worms versus when they're both kind of in the same spot. Because my thought was if you just have to kind of cope with whatever conditions are happening in the place where you're in, then you might essentially have a bigger array of things you secrete versus if a male can move around, maybe he can avoid some of the host immune responses a little bit more. So I thought that was a really interesting aspect of biology.
Gina, that's a fantastic question. We haven't done the Onchocerca volvulus, which is the one that travels. And the reason being that Onchocerca cannot be cultured. So there is no life cycle to get Onchocerca. The only way to get it is to actually dissect these nodules. And we need, live worms to do that. And so any nodule that has been dissected is actually in freezers. What we did do, and actually that was in collaboration with the LPD, the NIH people, is that we thought, if the males are secreting this at such a high level, can we detect it in the blood of humans and in the blood of, humans infected with Onchocerca volvulus or infected with, Brugia malayi? And actually we can't detect it in the blood. We did 80 individuals, targeted metabolomics, and we just couldn't see it, unfortunately. But that's something I would love to do. And so working with the LPD people, they do have sites in Africa where we may be able to work with them. And I think that is like the question. I love that.
Okay, Mark, you're next.
Wonderful talk.
Thank you, Mark.
So I'm a yeast guy, and there the differential glycosylation is about agglutination between male and females. And so I was wondering if any of the differential glycome had to do with binding between males and females, since they stick together in this pocket.
So you're saying that they don't necessarily have different sugars, it's how they're binding or binding on?
Well, they're different proteins that then get glycosylated in different patterns. But what happens is males stick to A's stick to
yeah. Oh, actually, that's such a good question. So in nature, I wonder, you're right, in situ, in the infection, they're together. Would the males stick to the females? Because they are wrapped around the females, and I don't know. So the way we did it is that The way the life cycle works, artificially, is that they infect these gerds, these gerbils, but they infect them peritonally. The peritonia, I can't say the word. Anyway, they basically inject in the belly, and it doesn't go through the lymphatics. And then they collect, different stages, and they give them to us. So they'll give us, separated males and separated females. Now, I wonder if the experiment to do, based on what you just said, is to put the males and females together in culture and see what we pull out of that. It'd be kind of interesting. Yeah,
it might really induce different patterns.
Yeah. That's a great idea. Thank you, Mark. Yeah.
Leslie had one, but I was going to ask one first, Elodie. The I guess Pichia expression is going to add some sugars to your proteins, but it's still probably not the same sugars that the worm's going to add.
Yep.
Do you have to then compare purified native protein, glycoprotein, to Pichia or to E. coli? And do you think it's the protein or the sugar that's making the difference?
Right. So here, I think what we'll do is only focus on the protein. It will be a different sugar. I'd like to run those on an array to see what kind of sugars we are getting on these specific proteins. And the sugars it won't be like the native protein because of the specific sugar that is added on by the worm, but at least it'll have some sugars that could have an effect. And so what we can do is an experiment where we cleave the sugar off from the native, the protein from the recombinantly expressed, and then compare with and without the Pichia sugar and see if it exacerbates the effect or not. And we can do that even with our, our glycan. And actually, I think Joe had done that experiment where he took the glycofractions, cut the sugars, and then looked at the effect. He's done like multiple experiments, but but that's a really good point, Steve. Absolutely.
Thanks. Leslie?
Thanks. That was a really great talk. Thank you. It's a topic I know very little about, and I was very surprised by the lifespan of the parasitic worms. And I was wondering how that compared to free living worms. And also, I don't know that much about the disease, so I guess that means it's not usually fatal. But I was wondering if the actual lifespan is caused by people dying, or is that their natural lifespan, or do they even know how long these things can live in, if they were just given the perfect care?
Yes, so the lifespan, so these worms don't kill, but they cause, high morbidity where people are disabled. And so people can die of old age. And so we do have an idea of how long these worms can live. But there's, a range. So they do know that Onchocerca can live, about 15 years, but something like Loa Loa can live, 20 years, and Brugia is on the lower end. Now, next to the free living nematodes, so C. elegans has a lifespan of, I think it's days or weeks, it's very short. However, there are a lot of studies being done on age and lifespan in C. elegans because they can target genes and then increase the lifespan by days, and it's like years, for parasitic worms. And so at some point we had looked at these, they have a whole bunch of these age-related genes in C. elegans, and we looked at whether there were orthologs in Brugia, and there were a few, but we were wondering about lifespan, whether, if you could, and actually we couldn't target genes in these worms for the longest time, but now there's one of our collaborators, Tom Yunash, who's at University of South Florida, who's developed a CRISPR system where you can now start to knock out some of the genes in these worms. So that will change a lot of things for the work, the genetic work we can do on these worms.
Fascinating. Thank you.
Thanks, Leslie.
Thank you, Elodie. I've seen lots of thank yous and great talk comments in the chat. You should check those out before you log off.
Great.
And
Thanks, guys.